Hi, everybody. My name is Ethan Perlstein. I'm the founder and CEO of Perlara and the co-founder and CEO of Maggie's Pearl. I'd like to tell you a story today entitled Individualized Drug Repurposing for the Long Tail of Genetic Diseases. And to be clear, this is a co-production of Maggie's Pearl, which is a joint venture between Perlara and Maggie's Cure. So this story starts with a little girl named Maggie, who is our PMM2 CDG pioneer. Maggie from Michigan has PMM2 CDG, the most common congenital disorder of glycosylation, affecting at least 1,000 patients globally, most of them kids like her. For almost 2 years now, let me start again. For over almost 2 years now, Maggie has been taking a repurposed drug called Epalrestat that we discovered together using simple patient avatars. The best part is the drug is working, not just in Maggie, but in another PMM2 patient, and we're going to take this drug into a phase 2/3 pivotal study very soon. We're just wrapping up our IND submission as we speak. All of that was made possible with Maggie's help as the pioneer, as the first PMM2 patient to take Epalrestat. And you see a picture here of Maggie at Mayo Clinic, January 29th, almost 2 years ago, 2020, receiving her first dose. You can see her sitting in the lap of Dr. Abe Morava Kozic, who is the principal investigator, who is also speaking in this forum from a clinical perspective about the Epalrestat story. I'm going to tell you how we actually did the science to arrive at this discovery. And you can see Holly over there on Maggie's left is her mom and is one of the founding partners of both Maggie's Cure and Maggie's Pearl. So let me first start with some real world evidence. Now, this is all going to be data that's now captured in a publication that was just published in the Annals of Neurology last month. And this represents here data from the first year. So what we're calling the real world evidence from the first year of Epalrestat treatment. And what you'll see in the beginning is Maggie before Epalrestat, 3 months before Epalrestat. And then you'll see at about midway point, it will switch over to Maggie after being 3 months on Epalrestat, but both sitting on the same couch. So that should be the visual cue here to relate the two videos. So I'm going to start here, and then I might just skip ahead at some point once you kind of get a sense for what Maggie's baseline was before Epalrestat, and then we can scoot ahead to when she started taking it. So you don't need to be a child neurologist, I think, to see the difference here. Before she was quite shaky, didn't really have the core stability and balance to maintain her posture, sitting posture for more than a second. And you can see kind of the difference here. Now she can sort of sit still, basically, and you could see some evidence of movement in her feet and her extremities, some of that jerkiness, but otherwise you can see sort of a night and day difference in terms of her core stability. So this was one of many different signs that something was happening, and these pieces of evidence started to mount in the weeks and months after Maggie started taking Epalrestat. I'm going to advance to the next sort of piece of real world evidence. So consistent with these gains in gross motor control, there was also demonstrated quite clear gains in fine motor control. So before Epalrestat, you can see in the coloring book that there really wasn't any coloring going on. But then after Epalrestat, Maggie, actually was quite able to draw within the lines. And there are many pieces of artwork like this that she's produced that speak to her vast improvements in fine motor control. Here's another example, November 2018, so 3 months before starting on Epalrestat. When given her name as a visual prompt, she could trace it, but again, only with the visual prompt. And then by August of 2020, so 7 months on Epalrestat, you can see without any prompt, she's able to draw out her name as legibly as my 6-year-old would have at the time. And here's another piece of real-world evidence. So this was a conversation, a snippet of a conversation that Holly, Maggie's mom, had with another mom with the rare disease kiddo. And this was just commenting on the ability to have conversations with her, and sort of a little recap of a conversation that was had between Maggie and her parents. And all of this mounting evidence suggested that Epalrestat was having an effect. It seemed to be the difference, between what was happening before she started taking it and then after she started taking it. So with that as the initial sort of appetizer to, get you interested in this story, now it gets even more fascinating, I think, because how did we discover Epalrestat? Well, we use what we call patient avatars. In this case, we really were able to take advantage of the classical model organism, yeast, Saccharomyces cerevisiae, budding yeast. Also take advantage of worms, C. elegans. And then our human validation step required only a fibroblast, because PMM2 is a so-called housekeeping gene. It's present in all cells and active in all cells because it's responsible for the first step in what's called N-linked glycosylation, which is a process whereby sugars get attached to proteins. And proteins very much need these sugars in order to function. And most of your proteome or proteins are actually glycosylated or have sugars attached to them. So what we were able to do is exploit the fact that from an evolutionary perspective, N-linked glycosylation, this process is incredibly conserved. So yeast do it, worms do it, to borrow from the Cole Porter song, and of course humans do it. And so what I'm talking about here is this enzymatic reaction. And in fact, the PMM2 dimer, it forms this dimer to come together to be enzymatically active. That is conserved between yeast and humans and all animals in between. If you get rid of PMM2 function entirely, animals do not survive. So life is not compatible without PMM2 function. So by definition, all PMM2 patients have some amount of residual enzyme activity. And the way that they lose enzyme activity depends on the mutation. So what I've highlighted here are different variants, and you can just see them doubled because you're looking at the actual dimer that's forming between two PMM2 monomers. And you can see these various amino acids are labeled, and they all have a different effect in terms of deteriorating or damaging the protein. Some of these mutations affect the stability of the protein. Some affect act like an Achilles heel, or they target the Achilles heel of the enzyme, which is in this case this catalytic glutamate residue, or excuse me, this catalytic arginine residue at 141. Maggie's genotype was the R141 and the E139, but most kiddos, PMM2 kiddos, have different combinations. But R141 tends to be recurring, one of the most common mutations, as well as this F119, this light blue cyan colored residue that sits at this interface between the two monomers. So each of these different variants has a different consequence in terms of how it breaks the function. And so we reasoned we needed to model each variant precisely in order to understand were there mechanisms of action that could rescue across all of these different variants, or whether they rescued only one variant or a few and not the others. So we were able to create this suite of patient avatars where the same variants were modeled across all of the systems, including, of course, patient-derived fibroblasts. And to go along as a companion with these yeast models, we developed a worm model of PMM2. And I'm just showing you here that we only really focused on one of the variants, the F119L, and we created this homozygous variant. So not to model any particular patient, although there are patients out there with this precise genotype, this, for a number of different reasons, was the most practical animal to construct of all the possible variants we could have. And we just show you here a graph that, and this is published from 2 years ago now in Disease Models and Mechanisms, we can show that in the orange plot, in the orange points, what we see here is a decrease in PMM2 activity compared to the control in blue, and then in gray, a different CDG mutant. This is a mutant that's lacking NGLY1, which causes a different CDG. So we created a bona fide PMM2 worm model. We could prove that it had this enzyme deficiency, and then we were able to also show that these animals had a sensitivity to proteasome stress. And this is not too surprising. We knew this from studying NGLY1, which is the other CDG I just mentioned, which acts as a control in that other experiment. But in this experiment, we're seeing data that's really informed by the NGLY1 experience. When we created NGLY1 worms, they happen to be extremely sensitive to bortezomib, which is a proteasome inhibitor. And when we tested our PMM2 worms, we also found that they were sensitive to bortezomib. And so that's what you see on the left if you treat these mutant worms with a wallop of this proteasome inhibitor. And then we screen for suppressors, which would then restore normal growth and development to these mutants. And so we were able to develop a high throughput screen in both yeast and worm models, where we were looking at the same outcome, which was, could we rescue the growth and development of these models? The yeast being unicellular, it could only do one thing, it could only grow, but worms going through developmental stages and being larvae and adults and creating offspring, you could model their entire life cycle, including their ability just to grow. And I'm going to skip over all of the results which have been published before, but this chemical screen ended up revealing a needle in a haystack. And it was this molecule on the left, which is an active component of cinnamon. And so this compound, alpha cyano-4-hydroxymic acid, is a natural product that we found was the best suppressor of our yeast and worm PMM2 models. And it turned out that Ono Pharmaceuticals, a Japanese pharmaceutical company, created a drug called an Aldose reductase inhibitor called Epalrestat, which is remarkably similar to the cinnamon compound. And this was an insight we made. I don't believe that the Ono pharmacologists or medicinal chemists were influenced by the cinnamon compound per se, but the inclusion of this carboxylic acid moiety is a known feature of aldose reductase inhibitors. And so the point was our yeast and worm models revealed that this class of carboxylic acid containing aldose reductase inhibitors was active in rescuing growth and developmental defects in PMM2 models. And we had not only this natural product that did this, but we also had a synthetic aldose reductase inhibitor that was approved, most critically, is approved for use in Asia for the treatment of diabetic neuropathy in adults. And I'll get to what the significance is and the connection between diabetic neuropathy in adults and a pediatric rare metabolic disorder involving a glycosylation defect. But here's the data that really tied together the cinnamon compound and the Epalrestat aldose reductase inhibitor, showing us across both worm and human fibroblast systems, where when we measured PMM2 activity, Epalrestat caused a modest increase, as did the cinnamon compound, abbreviated CHCA. And again, we were able to show that this modest increase was conserved between both worms and fibroblasts. So this was the novel discovery summarized in the disease and models and mechanism paper. And from this result, we got very curious to then test other PMM2 patient fibroblasts, because we want to understand, well, what is the significance of a 30 to 40% increase in PMM2 enzyme activity? Was that going to be a lot? Was that going to be a little? Would it be therapeutically beneficial? So in a paper that was recently published, as I mentioned earlier last month, in the Annals of Neurology, when inspecting 10 different patient fibroblast lines, you're looking here, and let's just focus on panel A on the left, you can see that the before and after the vehicle or the DMSO treatment versus the Epalrestat treatment, you can see that the PMM2 enzyme activity is going up in almost every case. There are two cases in two patient samples where either there's a flat response or a slight decrease, but in all the other cases, there was an increase in PMM2 activity, and it was objectively, quote unquote, small or modest. And when we looked at conventional glycosylation markers like ICAM1 and LAMP2, we didn't really see what appeared to be a clear and consistent increase. We could see increase in ICAM1 expression, but that was based on mRNA, not based on the protein itself. So we would infer that there was full-length glycoproteins produced. But these two conventional classical glycoprotein markers really didn't show that there was a functional increase in glycosylation. So we turned to an unbiased approach where we inspected the entire glycoproteome. And this is, again, this should say accepted. This is, I should have updated this citation here, but yeah, this was accepted and published last month. But what we showed in this more unbiased omics or glycoproteomics specifically approach is that you could see a essential complete rescue, near complete rescue of the glycoproteome. So this is sampling many individual glycoproteins and inspecting the glycosylation status of each of the glycans at each of the glycosylation sites. And then what you see is that Epalrestat in figure D, you see in this volcano plot that this is the comparison of Epalrestat treated PM2 fibroblasts versus untreated. And we can see here now this shift of the plot all the way to the right, whereas it had been at steady state shifted to the left. So this comparison in B is the untreated PMM2 fibroblasts compared to wild type control. So the hypoglycosylation phenotype across many glycoproteins was rescued by Epalrestat. And in fact, when we later inspected LAMP2, we could find that indeed there were increases in glycosylation by this method, but using Western blotting and mRNA expression in fibroblasts was not the perfect way actually to reveal the effects of Epalrestat. And I should note is that there were even patients whose PMM2 activity did not increase much with Epalrestat, but their glycoproteome was rescued. So we have some sort of caveats to the fibroblast Epalrestat responsiveness phenotype and how predictive it is of actual functional rescue. We think these glycoproteomic measures are much more accurate, and they reveal that Epalrestat really has a powerful functional rescue effect across the glycoproteome. Then we started to think about how could we validate this molecule in an actual patient. We have great data from worms and yeast and fibroblasts, but what would it all mean if it didn't do anything in an actual patient with PMM2? So Epalrestat is an approved medicine. It was approved in Japan, so we were able to take advantage of its multi-decade-long safety record and understanding of its pharmacokinetics and other properties, and begin an N-of-1 study with Maggie. But I want to show you here data that is the published data on Epalrestat and its preclinical data package, but also what it's doing in adults, because that set the expectation of what it would do in a child, because no child had ever been given Epalrestat before, that of course that we're aware of. So this is an example of the fact that Epalrestat clears relatively quickly in the blood, and that there's a longer retention in after eating. And we can sort of see, we didn't model the pre and post eating, but you can see there, this is data coming from Maggie's N-of-1, which is now again published. Forgive me for not having updated this citation here, but this is published. You can see here that the Epalrestat is cleared with similar pharmacokinetics in Maggie as it is based on all the experience in adults who have been taking this medicine for decades. So that's reassuring to see that this drug behaves similarly, at least in Maggie. We can also see here that the intended effect of Epalrestat, one way to measure it is to measure the amount of sorbitol in urine. Sorbitol is elevated in the urine of patients of diabetic neuropathy, and that's what's driving the mechanism here of peripheral neuropathy, which is what this drug was prescribed for. And so as expected, you can see, so that the left is just showing the table of the elevation at baseline of patients with diabetes of sorbitol relative to non-diabetic patients. And in the table on the right, you can see that Epalrestat can cut that sorbitol increase in half and essentially act to normalize. So we have that expectation that Epalrestat would do the same thing in Maggie, and indeed it does. So the two interesting results here were that just like adults with diabetic neuropathy, Maggie, and then it later turned out upon investigation of other children, the majority of kiddos with PMM2 actually have elevated urine polyol, specifically elevated sorbitol and elevated mannitol. And you can see here that 2 time points after treatment with Epalrestat, 3 months and 6 months, that sorbitol and mannitol urine levels were normalized by Epalrestat. And this is, again, part of this larger study, this larger consortium, the FCDGC, where a number of, I believe, 24 different patients with PMM2, their urine sorbitol and mannitol levels were assessed and then compared to neuropathy scores and liver pathology scores. And you can see here, especially with neuropathy, as we expected and as predicted from what's going on in adults with diabetic neuropathy, we can see here a correlation with the higher, the more severe the neuropathy, the more sorbitol accumulation at baseline. That signal is present with mannitol, but weaker. So it's really a pronounced signal with sorbitol. And this is all published as well. And so we were able to show that in addition to all the real world evidence, we showed quantitatively the effects of epalrestat. So here are two outcome measures: weight and transferrin glycosylation status. So transferrin is a protein produced in the liver that gets glycosylated normally at a very stereotypical number of sites. And in PMM2 patients and in type 1 CDG patients, generally there is a hypoglycosylation or an underglycosylation of transferrin, which you can detect here with this mono oligo to di oligo ratio. And you can see that in the time before Maggie started on epalrestat, her transferrin levels were bouncing around in the abnormal range. And then within 6 months of taking epalrestat, her transferrin, liver transferrin normalized. And there was a blip here, which we believe related to the fact that she started to gain weight and was still at the same dose, however. And so we believe here that the transferrin was actually a marker of target engagement. In addition to the sorbitol being the direct one, this is a functional readout that is sort of like the glycoproteomics analysis, except this is obviously one protein. But you can see here that this protein appears to be responsive to her weight and her dose. And so there was a period of underdosing here as her BMI sort of rocketed up. And then once it stabilized, then an increase in the dose was able to renormalize her transferrin. This is looking at her ICARS score, which is a measure of ataxia. It's an international ataxia rating scale. You can see in the dark blue point, she was on another medicine called acetazolamide, which is also being repurposed for PMM2, but it does not address the root cause of the disease. It does not increase PMM2 activity, but it does have effects on ataxia. And so you can see that when Maggie was on epalrestat for a few months, her ICARS scores improved. And in this rating system, lower numbers are better. She discontinued acetazolamide in order to start taking epalrestat for 30 days. She was rebaselined here, and you can see that her progress continued. And this is notable because in a previous study of acetazolamide treated kids, when they were taken off of acetazolamide, they essentially lost all of their gains within a few months. And what we see here instead is that Maggie continued on the same trajectory. And so again, speaking to the fact that epalrestat has functional impact, and that all the biochemical labs readouts we were seeing were translating and consistent with the real world evidence, and then with this one quantitative rating scale. And then the other question of safety, we knew that this medicine has been given and prescribed to millions of patients with diabetic neuropathy across Asia, not just Japan actually, but also India and China as well. And epalrestat, again, was approved in 1992 for the first time. So we really do have decades worth of safety data and track record on this compound, even though this is the first time it's going into children. And of course, children with an inherited metabolic condition. But you can see here that even with dose escalation within Maggie's treatment course, her ALT and AST, two measures of her liver enzyme, or two liver enzymes that are a measure of liver health, are essentially just moving sideways and are not importantly going up. And now I'll conclude with some data from a second child. His name is Ethan as well, and he's from Canada. And I'll show you, Ethan, so Maggie will be on epalrestat 1 year at the end of January. Ethan just crossed his 1 year mark on the drug. And what we found, especially when it comes to motor, gross motor, Ethan already had a higher baseline than Maggie before epalrestat, but we saw the same types of improvements as I'll show you in these videos here. So here's on the left, 3 months on epalrestat, and I should note that these are Ethan's sort of first unassisted steps here. And then on the right, we have 4 months. So a little bit farther. No, Dad's just sitting back a pace or two farther this time. And let's go to 5 months. So again, Dad's standing back a few paces even further. And then he shows off a little bit there with the jazz hands. And then I think kind of most impressively, here he is walking up an incline. And this was from the past summer. Not bad, if you ask me, and hopefully at some point he'll be doing this without the walker. And he's again only on year 1, and I don't have video here yet that's been compiled, but if you go to YouTube, you can search for Holly Carmichael and Maggie as well, and you can find a walking compilation where just last month she took sort of her first steps with assistance back and forth across her living room. So that's after 2 years on the drug. This is Ethan, who had a higher baseline after 1 year. So I suspect in a year's time next summer, there will be even more progress to report. So we're excited that these 2 N of 1 studies have, we think, provided the proof of concept, the clinical proof of concept that justifies scaling up Epalrestat and treating more PMM2 kids. And we are on the threshold with Dr. Marava Kozic and the Mayo Clinic of launching such a phase 2/3 pivotal study in the new year, hopefully as soon as January. We're wrapping up our IND submission as we speak, and will be submitted this month. So if you want to learn more about Epalrestat, about the trial, about anything I discussed, please feel free to either email me, check out maggiespearl.co. You can even check out perlara.com. There's information there as well. But I'm happy to receive any solicitations or questions or comments or feedback, especially if you are a PMM2 family and you want to learn more about the trial. Definitely check out maggiespearl.co and email me if you have any questions. Thank you for your time and attention.